Hi, my name is Pini Perera, and I'm a research assistant professor of environmental health sciences at the University of Michigan School of Public Health. Together with Doctors Dolonoy, Goodrich, and Svoboda, we have formed an environmental epigenetics lab to understand how environment could influence the epigenome. So, what is epigenetics? Epigenetics refers to the changes in gene expression that occur without altering the underlying genetic code. The epigenetic stimulus can drive the production of RNA from DNA. As more RNA transcripts are produced, more protein is generated. So the amount of gene expression is not dependent on the DNA sequence itself, but rather the epigenetic stimulus, which could be composed of modifications such as DNA methylation, histone modifications, or non-coding RNA. During my doctoral training in Dr. Jumyoung Kim's lab at the Louisiana State University, we used mouse knockout studies to investigate an epigenetically regulated mechanism called genomic imprinting. The knockout mice were smaller in size with problems in feeding. We also found that genomic imprinting is a tightly regulated process with tissue, sex, and developmental stage specificity. During my postdoctoral training in Dr. Dana Dolonoy's lab at the University of Michigan, my research focused on how the environmental exposures could disrupt the epigenome, with a particular focus on non-coding RNA called PIWI interacting RNA, also known as piRNA. We found that piRNA is expressed from somatic tissues, including the brain, heart, liver, and kidney, and that piRNA expression is affected by perinatal exposure to lead, particularly in the male mouse brain. My current research program aims at understanding how early life exposures impact health. To address this question, I use mouse exposure models coupled with molecular biology techniques such as quantitative RT-PCR to measure gene expression, small RNA sequencing to measure piRNA expression, and pyrosequencing methods to measure DNA methylation upon environmental exposures. So, as we know more about how environmental exposures impact gene regulation and long term health, we can find ways to either identify diseases early using epigenetic biomarkers, or we could also identify potential interventions through epigenetic mechanisms like piRNA. We at the University of Michigan School of Public Health promote diversity, innovative research, and community engagement. Thank you for listening, and please reach out to me if you have any questions.